Okay, so we've got maybe 6 or 7 slides to finish up here for this week, and the majority of this is going to be about the nucleus and a series of internal membrane structures inside the cell called the endomembrane system. So let's just start off with a quick overview using this animal cell diagram and this plant cell diagram. So the nucleus obviously is this structure here in the animal cell and this structure here in the plant cell. Now, one thing it's good to be aware of as you look at these diagrams is that not only are they models, as we talked about on Wednesday, but they're drawn here in 3 dimensions, and I would discourage all of us from trying to draw in 3 dimensions because it's very, very difficult to do, and we often don't capture the structures as we want to in a model representation of an actual cell. So that is the nucleus, and then the endomembrane system is composed of the following components: the nuclear envelope, the endoplasmic reticulum, the Golgi apparatus, and the plasma membrane. And so those structures represent the endomembrane system. And then the lysosomes, the peroxisomes, and vacuoles, if they're present, they're really kind of ancillary parts of the endomembrane system. But the parts that we really want to focus on in this discussion are the nuclear envelope, the endoplasmic reticulum, the Golgi, and the plasma membrane. So that's what we'll focus on here. And so in these diagrams, the components of the endomembrane system are highlighted kind of in purple and in blue and in a kind of light shade of pink. So that gives you a quick overview of the nucleus and the endomembrane system. Now let's kind of take a look at the first structure that we have to in these last few slides, the nucleus. So the nucleus is obviously the place where we find the DNA in the cell, and so the DNA in cells is composed of both the molecule called DNA, but it is complexed with proteins, and together this complex of DNA and proteins is called chromatin, and so that's what you find inside the nucleus. And we'll talk more about chromatin when we get to DNA replication and chromosomal structure. So that's inside the nucleus, and the nucleus is surrounded by a so-called double membrane that make up a structure called the nuclear envelope. So the nuclear envelope down here has 2 membranes, and you can see that they kind of loop back on themselves, and that's what gives us this double membrane structure. So if I draw this out, and it'll take me a little time to draw it out, if we're thinking about a double membrane, is what we're really thinking about is kind of you got to imagine this wrapping around and so that would then kind of wrap around to make one of these kind of looped membrane structures. And so this is really just a series of sheets, and within the sheets are these small holes, and those are called nuclear pores, and we'll take a look at a nuclear pore in a bit more detail in a moment. So this structure we call the double membrane of the nuclear envelope, and so don't forget this is a representation of a phospholipid, and so we've got a leaflet of phospholipids here and a leaflet of phospholipids here, and they create this double membrane structure that contains the genetic material. Within the nucleus, there is a poorly organized structure called the nucleolus. We're not going to worry about that too much. Just be aware that it's there. It is not membrane bound. It's just a discrete region where some specific functions occur in the nucleus. But we're not going to worry about that. You'll talk about that in cell biology when you get to that. So if we then take a look at one of these nuclear pores, one of these structures that is kind of like a gap in the nuclear envelope. If we take a look at that down here, just to orient ourselves, here is the nuclear envelope right there. And then the nuclear pore is not just a hole, it's a structure made of proteins that controls the movement of material both into and out of the nucleus. And this will become really important when we start looking at DNA replication and transcription and even translation. There are certain enzymes which have to work inside the nucleus. So enzymes that are needed inside the nucleus in here, if they're made out here in the cytosol, then they've got to get in. And so trying to get those molecules in requires regulation through this gate called the nuclear envelope. And so if we're trying to get a protein in from out here into the nucleus, it's got to come through this gate. And similarly, if there's a protein in here that needs to come out, it's got to come out through this gate so it can go into the cytosol. And so that's called a nuclear pore complex. And that's a group of proteins which simply acts as a gateway controlling what comes into and what leaves the nucleus, the inside of the nucleus. So let's start now talking about the components of the endomembrane system. So just remember that the endomembrane system is the nuclear envelope. It is the endoplasmic reticulum, which we abbreviate to the ER. It is the Golgi, and it is also the plasma membrane. So collectively, those structures make up the endomembrane system. And so we're going to start off just by taking a look at the endoplasmic reticulum. So the endoplasmic reticulum, as you can kind of see in this diagram, this little diagram up here, the endoplasmic reticulum is kind of continuous with the nuclear envelope. So the leaflet of the nuclear envelope is kind of has bulged out here and has become the structure we call the endoplasmic reticulum. And so you see that at lots of places. It's not shown extensively here, but you get these sheets of membrane that kind of originate from the nuclear envelope. And there are two types of endoplasmic reticulum. There is rough endoplasmic reticulum, and there is smooth endoplasmic reticulum. So the rough endoplasmic reticulum is characterized by what is shown here in this model as these little dots. And these little dots are actually ribosomes. So when we talked about the function of the cytosol, I said that one of the functions of the cytosol was translation. But I said that was one place in the cell that you get translation. And here is another place in the cell where you get translation. So on the endoplasmic reticulum where you find these ribosomes, that's where you will find translation occurring. So what happens here is that a ribosome, which I'll just kind of draw just kind of like this, kind of looks like a ribosome. So a ribosome, when it starts translating, sometimes it will get told that it has to go and translate at the endoplasmic reticulum. So there's a signal which tells this ribosome to come to the endoplasmic reticulum. And when it gets to the endoplasmic reticulum, it docks on the endoplasmic reticulum, and it will translate its protein into the inside of the endoplasmic reticulum. And so proteins that are made inside the endoplasmic reticulum, and we call that space inside the ER the lumen. So proteins which are translated from ribosomes on the ER and end up in the lumen, that's this space that I'm coloring in red here, those proteins are translated in there because they have to be specially modified by enzymes that you only find in the endoplasmic reticulum lumen. So the main chemical modification that we find occurring in the endoplasmic reticulum lumen is a chemical modification called glycosylation. And that literally means the addition of sugars. So proteins which get translated on ribosomes at the endoplasmic reticulum, so the rough endoplasmic reticulum, when they go into the lumen of the endoplasmic reticulum, they have a whole range of different sugars, monosaccharides, disaccharides, added to certain amino acids in those proteins. And so you kind of get a hybrid macromolecule there. It's kind of it's a protein, obviously, but it's got a sugar added to it. And that sugar basically acts as a signaling molecule, which directs a protein from the inside of the endoplasmic reticulum to other locations in the cell, or possibly even outside of the cell. So it's like a targeting molecule. So proteins that need to be kind of sent to various places in the cell have to be translated into the endoplasmic reticulum, or some of them do anyway. So that's the rough endoplasmic reticulum. And as you can kind of see, you tend to find rough endoplasmic reticulum closer. Well, this is rough endoplasmic reticulum. Tend to find it closer to the surface of the nucleus. And then all of the endoplasmic reticulum that we find out further away from the surface of the nucleus, that is smooth endoplasmic reticulum. Now, the only difference between the two really are the enzymes that you find inside the lumen, so inside the endoplasmic reticulum. So the enzymes in here, I'm coloring in green, are going to be different than the enzymes you found in the ER that is closer to the nucleus, which is the rough endoplasmic reticulum. So what does this endoplasmic reticulum do? And this endoplasmic reticulum, the smooth endoplasmic reticulum, has got several functions. For example, one of its functions is to store calcium. And calcium is a very important signaling molecule, and we'll talk about calcium signaling when we get to the cell communication and cell signaling part of the curriculum. The other important role of this part of the endoplasmic reticulum is detoxification. So certain toxic compounds which enter your body can be transported into the endoplasmic reticulum, the smooth ER, and there they can be detoxified. So in organs, the cells of organs that you find having a role, important role in detoxification, such as in your liver, for example, you'll find a lot of smooth endoplasmic reticulum. So that is our endoplasmic reticulum. Now, let's just take a look at the endoplasmic reticulum as if we were looking at it through an electron micrograph. So here is a diagram, or here's a colorized image from what's called a transmission electron microscope. And you don't need to worry about how this works. Just be aware that this is a type of very, very sensitive, high-resolution microscopy. And so is what we have is up here, that's our double membrane of our nucleus. So there up here is our nucleus. And then we have this is our rough endoplasmic reticulum. There's a sheet or a strand. And these are kind of like, stacks of membrane. And all these red dots that you see, I'll circle a few in green because that seems to be a good contrast against the red. These are the ribosomes that you find on the rough endoplasmic reticulum. Now, there's not a lot of smooth endoplasmic reticulum, but there's a little bit that you can see. So this would be smooth endoplasmic reticulum. There's a little ribosome there, but now there's not a lot of smooth endoplasmic reticulum here, so that's a little bit more difficult to see. But this gives you an idea of the organization of the endoplasmic reticulum in the cell, and you can kind of see how it branches here. There's a big sheet that branches out around here. So these are highly branched sheets of membrane that you find close to the nucleus, and then just down here there's a mitochondria kind of lurking off in the corner down here. Now I've just drawn over the scale bar, and I don't want to do that. I want to clear that off so that I can show you the scale down here so you get an idea. That's 0.1 micrometers, and so that is the same as 100 nanometers. So if you remember, if we go from 1 meter divided by 1,000, that is 1 millimeter. And if we take 1 millimeter and we divide that by 1,000, that is our 1 micrometer. And we're looking at this little bar here. That is 1/10 or 100 nanometers. So 1/10 of a micrometer is the same as 100 nanometers. So 100 nanometers is equal to 0.1 micrometers. So that gives you an idea of the scale of these things and what they look like down an electron micrograph. Now, the endoplasmic reticulum is kind of functionally connected to the structure called the endoplasmic reticulum. That's Eddie and Henry banging, so I'm going to pause this a moment. Okay, they finished their barking at the mailman, so we can carry on now. So as I was saying, the endoplasmic reticulum, which is kind of shown diagrammed here, is functionally very, very, very tightly connected to the endoplasmic, to this structure here called the Golgi apparatus. So to understand really the function of the Golgi apparatus, we've got to keep thinking about the endoplasmic reticulum. So the Golgi apparatus is kind of like FedEx or UPS, if you don't like FedEx and you think about UPS. And what I mean by that is that the Golgi will take material from the endoplasmic reticulum and it will send that material to other places in the cell or maybe even outside the cell. So when I say material, what that means is really proteins and lipids that are made in the rough ER and also in the smooth ER. So when one of these ribosomes on the surface of the endoplasmic reticulum, the rough endoplasmic reticulum, when it translates a protein into the inside or into the lumen of the endoplasmic reticulum, that protein is being translated into there. It's being made inside the ER lumen so that it can be sent to somewhere else in the cell. So imagine that there's a protein that is translated into this piece of endoplasmic reticulum here. The way that we send it to another place in the cell is that a little piece of the membrane of the ER kind of pinches off. And so anything inside the ER ends up being pinched off into one of these little kind of vesicles, which is just a little spherical kind of parcel of membrane. And so that's called budding. And then this little vesicle, or maybe this little vesicle here, inside it is protein and lipid that was produced at the endoplasmic reticulum. We're just going to call that the cargo. And so this vesicle comes along, and what it will do is it will kind of fuse with this structure called the Golgi. And there's a nice little example of a vesicle here, and you can see how the vesicle is fusing with this part of the Golgi. So what is the Golgi? The Golgi is a stack of membranes, much like the ER is a stack of membranes. And so there's kind of like a pancake of membranes. There's another kind of sheet. There's another sheet. And these sheets are called cisternae. Okay, so that's the formal name. And here's another sheet here. And then there's another sheet here. So there could be, anywhere from 5 to 6 to 10 to 20 of these kind of stacks or these cisternae. And we break the stack of cisternae down into regions. And the region closest to the endoplasmic reticulum is called the cis Golgi. Then the piece in the middle is called the medial cisternae. And then we have the trans cisternae, which is closest to the outside of the cell. So this over here is kind of the ER side of things. And then this side is the kind of membrane cell surface side of the stack. So what we've got, remember, is we've got these kind of components, these proteins, these cargoes, these lipids, inside these little vesicles which are budded off from the endoplasmic reticulum. And they get kind of deposited inside these stacks of membrane. And in that stack of membrane, there are more enzymes which modify chemically the proteins and the lipids. And again, we're dealing with the processes of glycosylation. So glycosylation is when we add sugars. And so in this Golgi apparatus, there are more sugars being added. And then remember, those sugars really are going to act as a signal for the protein to be told by the cell where it should be going in the cell. And so let's imagine, let's just think about a protein, a specific protein. Let's think about the protein insulin, which is produced on ribosomes on the rough endoplasmic reticulum. So it gets translated into the ER lumen, and then it ends up in these little. It gets translated into these lumen of these endoplasmic reticulum cisternae, and it gets packaged into these little vesicles. Then these vesicles fuse with these stacks of membrane on the cis face of the Golgi, and then what happens is another little vesicle will bud off, and inside that will be the cargo, and then it will kind of fuse to the next stack of membrane, and then another one will bud off and fuse with the next stack. And so the proteins kind of move through the stack, getting progressively more glycosylated. And once they've got a particular pattern of sugars added to them, that signals that a protein such as insulin can be excreted from the cell. So in this transcisternal stack here, all these proteins in here, and this little vesicle is about to bud off, and in here too. So inside here and inside here, those could be specific insulin molecules. And this vesicle here knows that it's got to fuse with the plasma membrane. And in doing so, it's going to release its cargo outside the cell. And here I'm just using one example where insulin would be released outside the cell. So the insulin is made kind of over here inside the lumen of the endoplasmic reticulum. It's packaged into these little vesicles, sent to the ER. It moves through the ER, getting sugars added, and those sugars basically tell this vesicle that your contents has got to be released outside the cell, and eventually that insulin will end up in the blood, where it can then regulate your blood sugar levels. And so there are lots and lots of examples of proteins that are made in your cells which have to be released into the environment. When I say environment, I mean the body's environment, so into tissues or into blood, for example, to be transported around the body. And so that's how you do this release of certain proteins. Now, sometimes you need to send proteins to do other functions in the cell. So rather than a protein being sent to be released, it will be produced into a little vesicle that will stay inside the cell. And so this little vesicle can be released. Maybe another vesicle down here will be released. And rather than fusing with the membrane like this one here did, they stay inside the cell. And those are typically called lysosomes. There are also little vesicles which can bud off of the endoplasmic reticulum as well, but we're not going to talk about those here. And so let's take a look at lysosomes. So lysosomes don't fuse with the membrane like these vesicles do. These are called secretory vesicles, and they're different in function than the lysosomes. So these lysosomes are for recycling materials inside the cell. So if you've got a mitochondria, for example, that needs to be broken down, then the mitochondria will be kind of engulfed by a lysosome, and the enzymes inside the lysosome will work to break down the mitochondria, and this can also be done on certain proteins. So inside those lysosomes are a bunch of proteins called acid hydrolases. Now, we talked about this word, so you know what this word means. This means that we're at a relatively high proton concentration, and so these enzymes, these hydrolases, that part of the word there should tell you that really that these enzymes are doing something in hydrolysis. And so what these acid hydrolases do is they take a molecule, and here we're just showing a molecule as having two parts, and R1 and R2. Now don't confuse these with R groups and amino acids. This is just to indicate two pieces of molecule. So it could be, okay, here is one amino acid joined to another amino acid, joined to another amino acid, joined to another amino acid. And I could say, okay, look, we're calling this piece here R1 and this piece here R2. And that's what these acid hydrolases do, is they break this bond here, so that peptide bond would be broken down by one of these acid hydrolases. And like you know, that's going to require some water. And is what we'd end up with is, in this case, 2 amino acids with a free H, 3 hydroxyl, and then we'd end up with another 2 amino acids. And so this would be R2, and this would be R1. And then we could repeat the process, and we could break these down even more if we added in another 2 water molecules. We could break those down even more. So this is the reverse of building up macromolecules. Now you remember when we built up macromolecules, is what we did is we took this monomer, let's say it's an amino acid, and we took this monomer, and we took an OH, and we took an H, and we joined those together to produce an H2O, and we ended up with 2 monomers joined together. And then if we wanted to add a third one on, we did the same thing. We pulled out this water molecule, and we ended up with 3 monomers joined together. And so what these acid hydrolases are doing is just reversing that. They're taking this water here, and they're putting it in, and they're breaking one of these bonds. And so here we're just saying that that's R1 and that's R2. And so we break that bond, and we end up with the 2 component pieces. So if we want to go backwards, we would just take that water molecule back out, and we would link those 2 things back up again. Okay, so this is the process we talked about when we talked about building things. And this process here is what you do if you break things down. And so it's these lysosomes which are working to break down molecules which need to be recycled because they've stopped working. So last thing to kind of talk about here is to revisit this idea of compartmentalization, because if you think about what we're doing here, if this is, okay, here's just a cell, here's a nucleus, that's good enough to be a nucleus, that's fine. If we want to do this kind of chemistry that we're talking about here, and we're doing it with enzymes that have to work in an acidic environment, we can't have the pH of the cytoplasm at say 5.6. That's not going to work, because there are lots of processes in the cytoplasm which need to work at pH say 7.2 or 7.4. So what we do instead is we have our endoplasmic reticulum. Here's my bad diagram of some endoplasmic reticulum. And we make our enzymes in the endoplasmic reticulum, which are going to be these acid hydrolases, and we put them in little vesicles, and we send them to our GOLGI. There's a GOLGI, and then they move through our GOLGI, getting modified and activated, and then they're released into a little sphere of membrane, and in there we can get the pH down to maybe 5.6, and that means that inside here these acid hydrolases can work. So there are 2 things going on here. The compartmentalization lets us create this acidic environment for this chemistry here to occur. We also don't want these hydrolases just kicking around in the cytoplasm, because if they were in the cytoplasm, they would just be breaking stuff down all the time. And so this is another one of the reasons that we create these compartments. And so the lysosome is just another example of a compartment where we can put certain enzymes, and we can use those enzymes to do certain biochemical processes. There's a lot of additional stuff. Don't forget what's on the test. That's in your learning outcomes. That's all I've got here. If you've got any questions, feel free to shoot me an email, put them in Skype, and I'll do my best to answer them. I will see you next week. Hope you have a great weekend.